Welcome back, welcome back, welcome back to science. All right, we're going to talk about the small intestine and the different segmentations of the small intestine. We're going to look at the cellular composition, how it works, how it absorbs, and the different things that are involved in that absorption. So, if you take the small intestine and you take it out of your body and line it up from end to end, you'll die. Anyway, it's quite a long organ in comparison to your small intestine, large intestine. All right. So the small intestine is going to start where the pyloric sphincter of the stomach ends. So at that moment, it's going to be the small intestine. Three different areas are going to be involved here. And if you look at where it is, this is where people are going to call their belly or their tummy as they're touching essentially where their belly button is, that area. So this is going to be most of what your abdominal cavity is. So you're going to have your liver is going to be a big part around the outside. You're going to have the large intestine, but in the middle, that's going to be your small intestine. So what does it do? It receives chyme from the stomach. The liver and the pancreatic secretions are also going to help in this process as digestion, even more of it, occurs here. And more importantly, absorption of those things that have been broken down. Whatever's left over goes to the large intestine. So, three parts of the small intestine. The first part is going to be called the duodenum. Duodenum is going to have it's the region that we've been talking about, how we've been identifying how the pancreaduodenal secretions occur, of course, in the duodenum. This is the shortest, and if you look at the movement of the others, this is the part that's really fixed, that doesn't move much. Moving from the duodenum, you get into the jejunum. So the jejunum is going to be thicker. It's going to move more. Um, and so in comparison of the three, it moves the most. The ileum is going to be after the jejunum and before you get to the point of being in the large intestine. So this is the most distal portion, and it's going to have the Peyer's patches that we talked about when we were discussing the lymphatic system. So the jejunum and ileum are suspended by a double layer. This to me kind of looks like an apron, and I've heard it called that anatomically. And so this apron is going to be the mesentery. And you can see that mesentery, a little bit of it here. And from here's a connection of the mesenteric, and you can see the artery and vein and the lymphatics that's going to be tied to there. And take a look here at this greater omentum. So this is that apron that's covering this. And if you go back, there's the greater omentum that's here, and you can see the lesser that's going to be towards the back, but it's still going to be there. All right. So this double folded layer of the peritoneum is going to be called the mesentery, and it transports blood and lymphatic vessels and nerves to the wall of the intestine. So the double fold, the uppermost part, that if you opened a person up and let's say a cadaver, the first thing you would see in the abdominal region after you got underneath the muscle is that greater omentum. It's going to be draped over the intestines. So just going through, here's the stomach, here's the pyloric sphincter, and now we move into the duodenum, and then we're going to get to the jejunum, and then we're going to eventually get to the point of the ileum, and we're going to terminate into the colon, specifically starting with the ascending colon. And take a look at that pretty little appendix right there. All right, this is a cross section, and you look at this and like, what is going on here? This is all the tubes that you see here at a cross section from the side. So that's why you see all those circles and odd looks. So yes, it kind of looks odd, but you can see some of the things that we haven't really discussed yet. And so let's see the parietal peritoneal. The visceral peritoneal is going to be actually on the organs. And that's no surprise. It's a theme that we've seen on every organ we've looked at. The viscera being on it like saran wrap, and that balloon nature where you put your fist into the balloon. Part of it is actually touching your fist. That's going to be the visceral. On the outer layer will be the parietal. And so you see this here in the abdominal cavity as well. So this is the peritoneal cavity. So you have the parietal peritoneal, which is going to be on the wall moving down. And the visceral peritoneal is going to be wrapped around every single thing that's on the interior wall of the material. All right, so let's take a look at the small intestinal wall. what's going on here. There are tiny little projections, and on this projection, there's even more projections. So these tiny projections of mucosa are going to be the intestinal villi. So everything about projections. So we're going to take a look here. Everything about this projection, and there's projections on the projections, just like all these microvilli, and they have those, um, all of these different villus with microvillus on top of it. Everything that is going to increase surface area. So think of my finger. My a mitten hand has this much surface area. Spread it apart a little bit, and you have surface area all the way around this. And if you took a measurement from like here to here, you get more like 8 to 12 inches. If you did this, you're going to have essentially way more, 24 inches, I would guess, that you would have the surface area to account for. And so there's lots of surface area that's extended because of those extensions. So surface area to volume ratio is drastically increased because of these microvilli. With all of those different absorption area that's there, surface area there, it's going to aid in absorption. So in each one of this villus, you're going to see a simple columnar epithelium at the core of the connective tissue, and at the center of that, you're going to have the blood vessels and the lacteal. If you remember this beautiful lacteal, the lacteal is this lymphatic vessel. It's the green in this picture. It's the Shrekish thing. And so you have that lacteal. And remember that the lymphatic system is a one-way system moving towards the heart. And at this point, this is where it's taking up the gastric fats, the fats that have been absorbed. And so it's really great that this is doing its job. It's going to be taking this. We know Peyer's patches that's going to be involved. So it's picking up the fat. That's where it's at. So the free surface of the epithelial cells contain the microvilli. So not only villi, but the microvilli as well. That also increases surface area. So we're talking about surface area on surface area, literally. And the intestinal glands, like the crypts of lubricum, they are located at the base of the villi. So you can see the intestinal glands, the lubricant that's there. The plicae circularis, these are going to be the muscular folds that are going to be found in the mucosa of small intestine. So the plicae circularis, you can see on maybe some different slides. Here we go. So if you looked at this, it's ridged along the inside. These are circular rings, and if you look at what they are, just I want you to focus on the surface area. It's such a beautiful thing. It is increasing the likelihood of absorption of nutrients. I mean, this is great. This is key for survival. If we were just a slick pipe in there, it would take so much intestines to do what our small amount of intestines do. I think it's 27 feet of small intestine. Anyway, we have these plicae circularis are these circular motions, circular structures, and you can also see there's circular muscle. That's the name of a muscle. You can see a longitudinal muscle, and you can see the serosa that's going to be on the outside. Once again, serosa, a common name for just an outside layer. So these ring-like regions are going to help, especially when we get to the talking about the point of peristaltic movements. All right, so we have the villus. We can see the different simple columnar epithelial that's going to be there, mucosal cells, the intestinal gland that's going to be there, and even further going from a lacteal to a lymph vessel, moving up that highway system. And here is the microvilli that's going to be on one of those cells. So these are additional levels of um, surface area. All right, so mucus. Mucus is going to be released by goblet cells. No surprise there. These are the columnar epithelial cells. That's kind of what they do, those goblet cells. They also have secretion, also secreted by the specialized Brunner glands. Um, the interkinase is where you may have remembered the word Brunner gland. And so the interkinases are going to be releasing interkinase, and interkinase is going to cause the chymotrypsin to become trypsin, And now the trypsin is going to have many different pathways that can be activated as the result of it doing its job. So this is going to be a thick alkaline secretion. And remember that alkaline, because the chyme is going to have that hydrochloric acid that's digested in the stomach. So now once it's there, we're going to have the stimulation like we need to have that bicarbonate to bring down that acidity. So we're not digesting our small intestine. We also have the release of watery fluids. Man, so this picks up digestive products and promotes them into the villi. No digestive enzymes in that watery fluid. It's just going to help the movement of things. It's going to keep things flowing, literally. So the enzymes that are going in the membranes of the microvilli, we have peptidases. These break down peptides into smaller amino acids. Like, okay, we have so many protein, right? We have so many protein things. It's a difficult compound. And we've had our proteases that have broken them down to these peptides, these polypeptides, and this is breaking it down into amino acids. Now that you have the amino acids, you can actually absorb those into your bloodstream and use them. Maybe store them in your liver to go through deaminization or the production of urea or whatever it may be. But we have the peptidases that are breaking down those final polypeptides into even amino acids. Other things doing their final digestive process. You have to think of it that there's been a giant products consumed that make nasty that you ate from the restaurant. You started digesting it when you saw that. You were releasing these fluids in your mouth, and then you started mixing the mechanical as well as mixing in the chemicals. Then you push that down, swallowing, soft palate up, epiglottis down. You're now swallowing, moving down through peristaltic waves through your esophagus, through the cardiac sphincter, and to the stomach, into the fundus, and then the body. Then you have the pylorus. You're moving it back and forth and churning it, moving it with the different muscular directions. Now that you have this chyme made, you're going to have a little bit leaking out into the duodenum, and you have the pancreatic juices as well as the bile from the common bile duct that's going to be released. And you're also going to have the releasing of bicarbonates to neutralize this, the alkaline mucus that's going to be there, the Brunner's glands that are going to be triggering the interkinases to cause the trypsin to be activated. So then we can have the different products that trypsin triggers to start. You've deconstructed things from something elaborate to now into subcategorical pieces. Now we have the final precision chopping. And so we have sucrase, which is breaking down sucrose into glucose. You have maltase breaking down maltose into glucose and fructose. Um, Long short of it is we are breaking down disaccharides into monosaccharides that we can actually use. Lipases going to break down fats. It's going to actually have the emulsification, and it's going to break down the fatty acids and the glycerol, so they're broken off. So we have all of these things that are happening, enabling your body to actually absorb here in the small intestine. So regulation of the small intestine secretion occurs by these methods. So these four. Mucus secretion occurs in response to mechanical stimulation and the presence of irritants such as gastric juice. So that's moving into your intestinal tract. So you deal with it. It's going to stimulate and cause like, okay, since this is here, we need to do this. So the contact with the chyme, it's going to stimulate goblet cells to secrete mucus and the intestinal glands to secrete the watery fluid. Once again, no enzymes there. The distension of the intestinal wall activates nerve plexuses in the wall of the small intestine, and distension also stimulates the parasympathetic, remember the rest and digest, to trigger the release of the intestinal enzymes to cause the breakdown of this to actually cause true um, chemical breakdown. So we can actually absorb this. Villi, they increase the absorption area. Small intestine is the most important absorbing organ in the alimentary canal. So that is the tube going from mouth all the way through. Because we're a tube in a tube, stuff, you can swallow a marble and defecate the marble out. So it's going through this tube in our body. Absorption is so effective that very little absorbable material reaches the organ's distal end. So very little things that you can actually absorb is defecated out. Most of it we go through, We strip every single part of a nutrient we can from it using all of these different kinds of chemical enzyme cocktails to break it down. So, carbohydrate digestion and secretion. Saliva and the pancreatic amylases, both are starch breaker downers, are going to break down starch and glycogen into disaccharides. The intestinal enzymes break down disaccharides into monosaccharides. The monosaccharides are going to be absorbed by facilitated diffusion and active transport into the blood vessels in the villi. And then we know we're going to utilize insulin to cause that to go into our cells. All right, so protein digestion. Pepsin in the stomach breaks down the proteins into polypeptides, and it takes the acidity of both the chief and the parietal cells doing their job, making an acidic environment, making the chymopepsin becoming actually usable form of pepsin if it's in the right pH. The pancreatic proteases, like trypsin and chymotrypsin, are going to break down proteins and polypeptides into smaller peptides. So once again, this is duodenal. So now after this, we're going to have the intestinal peptidases that are going to break down the peptides into amino acids. That's absorbable. So we're going to absorb that, put it in our blood vessels, and it's going to be absorbed through the villi. So this is the breakdown of maltose. And in the end, you have glucose. That's what maltose is. It's a disaccharide of glucose. And you can see at the bottom a dipeptide broken down into the different amino acids that are there. And they're just using generic versions to show in our group, just to show that dipepsidase breaking it down. All right, so fat absorption. So we went through carbohydrates, we've went through protein, now we get to fat. Fat digestion and absorption. We're going to have the emulsification because of bile salts. That's why if you're missing your gallbladder, you can't have that digestion, and you're going to have to use a low fatty diet and also take medication to help fix that. Digested mainly by enzymes from the pancreas and the small intestine. Digested into glycerol and fatty acids. We've broken it down. And now that the fatty acids and glycerol are absorbed by a process involving several steps, absorbed into the blood or lymphatic capillaries, the lacteals that we looked at. All right, so myocyte. What is a myocyte? A myocyte is going to have a nonpolar structure, and you're going to have nonpolar tails pointing towards it, and you're going to have polar heads wrapping around it. So you make a sphere of polar outside with a nonpolar center. Think of, um, to me, it's like a dandelion where the white fuzzy part, let's say a dandelion is now a sphere instead of just part of that flower. Um, the outer whiter part would represent polar things. The stem coming off to the seed that connects to that core would be nonpolar, and that core would be nonpolar. All right, so micelles are going to be loose complexes of fatty acids and bile salts, which from which fatty acids migrate to the microvilli and then can be absorbed. So the fatty acids plus the glycerol are resynthesized in the triglycerides in the smoothie R. We know that this is where we're going to make phospholipids and different things like that, like to make our cell membrane. New triglycerides clusters are encased in protein, forming chylomicrons. That's a great name for a cat. If you're trying to think of a good name for a cat, chylomicron is a good starting point. So chylomicrons can be absorbed by lacteals, and this is going to go into the lymphatic system. And we know ultimately that these chylomicrons, the fatty material, will eventually be dumped into your right or left subclavian, and then dumped into the superior vena cava. So this is showing the fat absorption, how it's going through smooth endoplasmic reticulum. It's going to alter it. It's going to make the chylomicrons. Chylomicrons are going to go out. They're going to be absorbed by that lacteal. Now it's going to be the lymph system that's going to then be moved later on. All right, movement of the small intestine. Small intestine is going to carry and move things in two ways. One is peristalsis. We know the balloon moving the marble down is peristalsis. Then segmentation is something we haven't talked about. Segmentation is going to have those rings where it's going to be essentially moving it back and forth, which is going to form a mixture. As it breaks apart and has more surfaces that are now exposed, you have more areas that can be broken down. So that's going to increase the effectiveness of the enzymes as it's working on it. The parasympathetic impulses stimulate both mixing and peristaltic movements. No surprise, because the parasympathetic is the rest and digest. The sympathetic is going to inhibit them. If small intestine becomes overdistended or irritated, a strong peristaltic rush is going to sweep contents into the large intestine. It's like, we're not dealing with this, just get it out of here. Get this crap out of here. And it does. It's called diarrhea. And please make sure you learn how to spell diarrhea, Diarrhea. Not a good animal, not a good name for a cat. It's a hard one to spell. It is the most misspelled word in the medical world. Diaphragm is the second most, Diaphragm. And thirdly, oddly enough, is alcohol. All right, so ileocecal sphincter. Ileo is referring to your ileum, and cecal, as in it's moving to your cecum, it's going to move to your large intestine. A sphincter is going to be a circular muscle. So if you had to use a little bit of deductive logic here, you would probably be right that this is a way that we regulate and how we're moving that material, that final breakdown stuff, into your large intestine. So this helps regulate the flow of chyme. All right, guys. Next is the great big bad large intestine. All right, I'll see you soon.